Hi and welcome back, everyone. I hope that by now you've had a chance to meet some of the forum's partners. Again, you can do so tomorrow if you have not. And I hope that at least some of you got to see the Millennium Pitching Contest final. In this session, we'll be hearing from the winners of the Millennium Technology Prize, announced last night, and we'll get an insight into their transformative work. Then we'll meet the awardees of the Millennium Youth Prize. So, to take care of all of those, I'd like to hand over to the chair of the session, the president of Aalto University, Ilkka Niemelä. Ilkka?
Millennium Technology Prize winners, Professor Shankar Balasubramanian and Professor David Klenerman, Your Excellencies, honored guests, ladies and gentlemen, I have the pleasure to welcome you all to the 2020 Technology Prize lecture. And it's also a great honor to be able to use this opportunity to congratulate you, Professor Balasubramanian and Professor Klenerman, on behalf of Aalto University for the Millennium Technology Prize. The prize is Finland's contribute to innovation for a better life. The prize highlights the extensive impact of science and innovation on society and people around the world. I'm pleased to say that this bold mission is wholly in line with the ambitions of Aalto University. When founded in 2010, the university was given a national mission to strengthen Finland's innovative capacity through first-class research, art, and education. We strive to build cutting-edge knowledge and technology so that we can offer solutions to the most pressing challenges faced by the society. These challenges call for education of professionals with knowledge and capabilities to build a sustainable future. With this in mind, at Aalto University, we strive to educate game changers of tomorrow. But to do so, we need to recognize that the change begins with children and young people. They are the courageous explorers and open-minded experimenters who shape the future. And we need to continue efforts to bring children and academia closer together. Aalto University has a long tradition of cooperating with schools to do just this, but we have gone further and take an internationally pioneering approach. The Aalto University Junior program expands the traditional STEM activities to include a unique combination of art, creativity, business and entrepreneurship, all under the same umbrella. Aalto University has also opened its campus to school students in an innovative way, and we have now 1,700 students on our campus in 3 schools. We at Aalto want to be a strong partner in the entire chain of education, enabling life-wide learning. Ladies and gentlemen, the Millennium Technology Prize is looking for innovations that will improve the life of our planet. Inspired by this, Aalto University and the Technology Academy Finland are organizing a competition for innovators for the age of 19 and younger for the 3rd time. Later today, we will award the Millennium Youth Prize to a talented young innovator who will briefly present her or his innovation in this event. I'm particularly happy that the winners of the Millennium Technology Prize are awarding the Millennium Youth Prize. You, professors Balasubramanian and Klenerman, are real role models for the young innovators. They can learn from you how determined work can lead to discoveries and innovations to benefit the world. But I think we are all now very eager to learn more about your groundbreaking technology. So it's my great pleasure to give the floor to the academic professor Päivi Törmä, the chair of the International Selection Committee, to introduce the 2020 Millennium Technology Prize winning innovation and the winners. Päivi, please, the floor is yours.
Ladies and gentlemen, I have the great honor to introduce Sir Professor Shankar Balasubramanian and Sir Professor David Klenerman. The Millennium Technology Prize 2020 was given to their seminal work on the Solexa Illumina Next Generation DNA Sequencing, NGS. This technique is based on fragmenting DNA, labeling it with fluorescent markers, obtaining information base by base, and combining with a computer. NGS has played a really crucial role in our fight against the COVID-19 virus. And it has been already applied in personalized medicine, for instance, cancer treatment. In the future, it will have a key role in sustainable food production and in preventing biodiversity loss. It is really revolutionary to be able to read and write the code of life, the DNA. NGS can be compared to some great steps in usual writing and reading. Namely, it's like going from stone tablets to papyrus paper. So obviously, there is still a lot to come. We are in the beginning of an exciting journey. Quite interestingly, Professors Balasubramanian and Klenerman told that the grant proposal that funded their breakthrough did not even contain the word sequencing. They came up with this application idea accidentally within a scientific adventure. And now, 20 years later, the fruit of their blue sky fundamental research is saving millions of lives. Professor Balasubramanian did his, he was born in Madras, India, and then did his PhD in University of Cambridge. After that, he had a postdoctoral stay in Penn State University, US, from where he returned to Cambridge, and he is there now Herschel Smith, a professor of medicinal chemistry. He, together with Professor Klenerman, founded the Solexa company that was started the development of NGS technique. But he has also founded another company, the Cambridge Epigenetics. Professor Klenerman was born in Wrexham, Wales, and also did his PhD in Cambridge. After that, he was a postdoc at Stanford University, then a researcher at BB Research Center, and then returned to academic career at Cambridge, where he is now the Royal Society Professor of Molecular Medicine. Professors Balasubramanian and Klenerman have received numerous national prizes, for instance, the Royal Medal that was awarded to them jointly. Now, we have the great pleasure to hear more about their great innovation directly from themselves.
Thank you very much for that kind introduction. We'd like to thank the Technology Academy of Finland for awarding us this prize and have the opportunity in this prize lecture to tell you a little bit about the technology we developed and how we came to develop it.
So we're going to tell you about the original ideas and proof of concept work that was carried out in the University of Cambridge. Then we started a company called Solexa in 1998, and it was the team at Solexa that reduced to practice the technology in an integrated first commercial system in 2006. Then in 2007, Illumina acquired the company and technology and continued advancing the technology to what it is today. And would like to acknowledge all the contributions from the university people, the Solexa team, and the team at Illumina. So we'll start with DNA, which is, of course, it's an information molecule, and DNA is composed of a linear string of 4 letters, G, C, T, and A. And it is the sequence in which how these letters are organized which provides all the information to make up genes in our genome, and genes encode for proteins which form the structures and machineries of cells and living organisms. And DNA it's fundamental to life, to all living systems. So in order to begin to understand what this information is and what it means, one needs to have a methodology for decoding or reading this sequence of letters.
So why sequence DNA? Like Shankar said, it's to understand life, and more importantly, to understand the differences between humans, because we all have a different sequence of bases. And if you could do that, you could find the causes of common and rare diseases. You could detect and diagnose these diseases. You could prevent people from getting these diseases. You could have the optimal treatment if they were unfortunate to get ill. And you can monitor the effectiveness of these treatments. But at the time we started working in this area, there was a problem. The human genome is very large. It has 3,000 million letters. And there was no technology that could determine the sequence of these letters cheaply, accurately, and rapidly. But back in 1995, can we have the next slide? We weren't thinking about sequencing DNA. Shankar and I had moved to Cambridge, and we got our first joint grant from the BBSRC. And the idea of that project was to watch an enzyme, DNA polymerase, in action, copying DNA. So what we were going to do is attach a single molecule of DNA to a surface. And you see this oval, that's the DNA polymerase. And we were going to watch it in real time, incorporating color-coded nucleotides into the growing strand of copied DNA.
So during the course of these experiments that were exploratory to try and understand the process of DNA synthesis from a different perspective, we of course had lots of other conversations, and shown in this photo here this is actually a pub just behind the chemistry department in Cambridge, and we used to spend time in places like this discussing the ideas of what we were doing, but also starting to see a bigger and different context for this. Now, in 1997, the International Human Genome Project was taking place in part quite near to Cambridge, about 15 miles away at the Wellcome Trust Sanger Institute. And there they were playing an important part in determining these 3 billion bases for the first look at what the human genome looks like. And during our conversations and brainstorming in places like the Panton Arms, we realized that what we were doing could actually be adapted to a method to read the code of a DNA strand. And so we started to think about, could we develop this into a method for sequencing human genomes? Because the Human Genome Project would sequence one genome, but we are, of course, all different. And to understand the genetic basis for what makes us all different and all the disease-related aspects that David alluded to, one would need to sequence lots of human genomes, and one would need a method that was much faster and much more affordable than the methodology at that time. So the concept for sequencing, we call it solid phase DNA sequencing. Some people call it Solexa sequencing now, is shown in this. It's an old schematic. So the DNA molecule you see has a long strand and a short strand. It's fixed to a surface. And you will see in the long strand, the first position where you would extend the short strand along has a T. Now, many years ago, the work of Rosalind Franklin, Francis Crick, and Jim Watson led to the discovery that G base pairs with C and A base pairs with T. So what we were thinking is, from our fluorescence color coding experiments, if you could color code each of the 4 building blocks, which you see here in green, blue, red, and magenta, if this machine called a polymerase puts in place the first correct building block, it will be the A, which has a red dye attached to it. So if we could control a single incorporation with A and then image that surface, we would know from the red color that it is an A that has gone in. Then if we could use chemistry to remove the color and remove some chemical groups blocking the second building block from going in, we could in the next cycle incorporate the next base, which would be a C opposite a G, and we would bring in the magenta color there. So every cycle you would see a series of colors changing, and by recording those color changes, you would record the sequence of that molecule of DNA on the surface. So why would sequencing on solid phase be helpful?
So the idea was that rather than sequencing one molecule, we'd sequence millions at the same time. So the way we were going to do this is extract the genomic DNA, fragment it and attach it to a surface. So we had single molecules of DNA, on average, just far enough apart that we could distinguish between them. And as I'll show you in the next slide, I did a simple calculation that showed that if we did everything in parallel, we could sequence much, much faster than the current technology. So the next slide shows you this calculation that I did back in August 1997. So the human genome has 3 billion bases, and I assumed that you had an array of 300 by 300 single molecules of DNA. And I assumed it took you 10 seconds to determine the color of the base that had been incorporated. This turns out to mean that you can sequence 10 to the 4, 10,000 bases per second, or 10 kilobases. So one machine would take you, we estimated, 5 days in order to sequence the human genome. So we saw this was a pathway to enable us to sequence the whole human genome of an individual very rapidly. So that was in 1997. This slide is in 2005 and is an image taken at the company Solexa that we co-founded. And it's this sequence of a small bacteriophage. And what you can see is all these different colored dots. These are molecules of DNA. So to achieve this data, we did exactly what I've described to you, apart from one important difference. Rather than studying a single molecule of DNA, we amplified the single molecule into a few hundred copies of the same sequence of DNA on the same place on the chip. And so we have these spots, red, blue, yellow, and green for the different bases. So 100 microns, that's the scale bar on the image, that's about the size of a human hair. So hopefully you can appreciate the high density of all these spots and the idea that we can sequence them all at the same time. If you look at the zoom, there's a 20 micron scale bar, and you can see these small amplified spots. Each one's about a micron. And this is an image for one round of sequencing. What we're going to do is read the bases in all these spots, then we're going to remove the fluorophore and repeat the sequencing cycle again, record the color of the dye incorporated at that spot, and we'll do that again and again. And in the next slide, Shankar will show you how we can reassemble that into the human genome.
So if at each spot on the chip you build up a sequence of bases, you end up with a sequence from very many fragments. Now, the Human Genome Project provided what we now call a reference sequence that tells you about the structure of a genome. So if you sequence fragments of DNA, you can use a computer to find out where that sequence came from, and you realign to the reference of the human genome. And by doing so, you build up your new genome. And you can start to see the differences between one genome and another. So today in practice on the latest systems that Illumina provide, you can read up to 250 bases from one end of the fragment and another 250 bases from the other end. So what does this achieve in terms of speed and capacity? Well, shown on the left is the first Solexa sequencing system. This is showing the instrument. It was called a genome analyzer. It was given the term 1G because it could sequence 1 billion bases of DNA in a single experimental run. And this was produced by the team at Solexa and released in 2006. To put that in perspective, one of these systems had a capacity that was about the capacity of the whole global capacity for sequencing around the time that we started the project. Now, shown on the right, 2021, is a picture of the latest systems that Illumina produce and sell. This is called a NovaSeq, and these can now sequence several trillion bases per run. That's several thousand billion bases per run. So this is a further 1,000 to 10,000-fold improvement on the first Solexa instrument. So what this means, put in perspective, is these instruments can sequence a genome roughly in about a genome an hour. The Human Genome Project took about 10 years and cost about $1 billion, several $1 billion. And today, a genome costs less than $1,000 US. So we're now going to move to an important part of the talk where we talk about applications. There are many applications in scientific research that we don't have time to discuss in detail today. We're going to focus on the applications to medicine. And our original motivation for this project was to enable population-scale human genome sequencing so that we could bring about the use of genomic information to help people. So the first example I'm going to talk through is cancer. Cancer, in its origins, is caused by changes to your genome, changes to your DNA. Now, many big projects that have used next-generation sequencing to sequence cancers and sequence cancer patients have been carried out. Some examples of the projects are shown on the left. There's the big international collaboration, the International Cancer Genome Consortium. And below that is the logo for Genomics England, which is actually a project involving the National Health Service in the UK. It's a taxpayer-funded project. And projects like these and research on the genomic aspects of cancers is starting to build up valuable databases of information on commonly occurring patterns of changes to your genome in certain cancers. Now, this information provides fundamental understanding on the cancers, but it can also enable cancers to be substratified. Shown in the middle is an example of a well-known mutation in a gene called BRAF. Now, this mutation occurs in about 50% of melanomas, skin cancers. And today, physicians prescribe a drug that targets the protein that BRAF encodes. And they get a very good response for tumors, melanomas that have this particular mutation. So it's an example of using the information from your genome and the genome of the tumor to prescribe a drug that will be more effective in that particular case. And as more and more information is provided, we will understand more and more about what is possible. So I predict over the next 2 decades, we will see a lot of advancement, but already clinicians are making use of data in this way. On the right, I want to say something about an area that's relatively in its early stages, but there's some very promising data already. It turns out that cancer cells release some of their DNA contents into the bloodstream. And so if you draw blood and you isolate this DNA that no longer has the cells associated with it, it's floating around, and you use NGS to sequence very deeply, it's possible to detect some of those patterns of mutations and changes that the cancer-related studies have shown are associated with cancers. So this is now being developed into tests where a blood test may be used to start to think about making a diagnosis of cancer, and there are actually already tests that certain organizations are releasing. Now, the direction of travel here, the hope, is that it will become possible in the future to detect at least some cancers relatively earlier than is possible today. And early detection of cancer is probably one of the most promising ways to ensure a better outcome, a survival outcome, to help patients.
So what I was going to discuss with you is rare genetic disease. Unfortunately, young children are sometimes born with poor development, and when they visit the doctor can't work out what's wrong with the child or come up with a treatment. So in this example, a child came to the doctor at 5 months with poor development, poor muscle tone, and suffering from seizures. So what they did is sequence the mother, the father, and the child and compared the DNA sequences. Initially, there were 22,000 differences, and they triaged these differences by asking different criteria. Did these differences code for proteins? Were they linked to disease? Were they damaging? And by going through those criteria, they reduced the differences down to a difference in one key gene that was associated with disrupted copper metabolism. And they could confirm a diagnosis of Menkes disease, and the doctor could then design a treatment for this child. So this took 4 days to do the sequencing and 10 minutes for the doctor to do the diagnosis. Now it's possible to do the sequencing in just over a day. So this is very rapid, and that's important in neonatal intensive care. And so this type of technology is now available in the UK at some of our NHS hospitals. And like Shankar said, for cancer, I'm sure there's going to be more of this type of use of this technology in the future.
The third and final application area we wanted to feature was infectious diseases. Once deemed to be a problem only for the developing world, but this is not correct. As we now know, infectious diseases is a problem for the whole world, all countries in the world. Infectious diseases are caused by pathogens, and pathogens have genomes. They're smaller genomes than human genomes. And one of the ways that NGS has been used is to take samples from patients who've tested positive or had symptoms of an infection, and to sequence samples and extract from that the sequence of the pathogen. Now, this has been used in many countries since the SARS-2 coronavirus pandemic started, in order to track the spread of variants of this virus. Shown on the right is an example of data from an international collaboration where data has been pooled. There are over 1 million cases of the pathogen SARS-2 coronavirus being sequenced. And this is allowing us on a global scale to track the emergence of new variants and the spreading of new variants. And this is being applied right now in countries to impose certain methods of management. The information will also be very helpful in thinking about the next generation of vaccines that may be administered in the future, customized to address the variants that need to be protected against. The other aspect of infectious diseases, and again the SARS-2 pandemic, is there are projects where patients who've been infected are having their genome sequenced. And the aim here is to try and understand from that information why some patients have a relatively benign infection, they're asymptomatic, whereas others end up on a ventilator having serious illness in hospitals. So this is really a taste of what can be done. It involves international collaboration and is enabled by sequencing.
So to finish, we'd just like to leave you with some reflections on the last 25 years and some important acknowledgements. So it's important to realize the idea came from basic science. So the message is, if you want the next breakthrough technology, please fund basic science. The other important message that this work required a talented team of people with different skills. I'm a physical chemist. I design ways of measuring molecules. Shankar is a chemical biologist. He makes molecules and understands how they work. And by working together, we could tackle a big problem. With the company, we had other skills that came in. We had engineers, we had people who could do the computing and bioinformatics. And it was only by having a talented team of people that we could working together that it was possible to develop this technology.
I would like to add a couple of reflections. This project started when we were relatively junior, and I think younger people in science are often more prepared to take risks than we do when we're more senior. And what I would say particularly to the young researchers out there is do take risk and try to identify and tackle big questions that you feel could make a big impact if you can. Opportunities like that they're relatively rare, but if you see them in what you're doing, I strongly encourage you to go for it and have a try. The worst thing that can happen is that you may fail, and then you try again. Last point, and again, there's been a lot of exposure of this over the last year with the development of vaccines and also tracking the spread of the virus. Scientists working together can solve major problems in society. I think this is very evident right now, and I think this is the way we all need to think about where science can go.
And lastly, we'd like to acknowledge our postdocs at the University of Cambridge and the BBSRC, who funded us initially to do the experiments on DNA polymerase.
And this was very much a project that was born in a university, led to a startup, Solexa, some fantastically talented people at Solexa. And again, I would like to salute them. And also the people at Illumina, who have made the technology what it is today and are continuing to develop that.
Thank you very much.
Thank you.
Thank you very much for your extremely inspiring talk. Now we have some questions in the chat, so I will ask you. The first one. Crick and Watson brainstormed in the Eagle Pub in Cambridge, and you two in the Panton Arms. Is there something about English beer that leads to innovation?
David, do you want to take that?
I don't think there's something about English beer. I like to say there was. But I think it's more the fact that we spent some time actually getting to know each other and talking to each other. That was the key thing. Yes,
thank you. Okay, then there's another one. Your bright idea became a publication, then a startup, and eventually a resource for humanity. What single factor about your institution or funders or even your own choices most helped in making it possible?
So I think it's more than one factor. I should say we had a lot of luck in this project from start to finish. We also had a lot of failures at every stage in the project that we didn't share with you. And I think working through the failures and learning from them and trying again somewhat differently is critical to scientific research, any problem, and this was no exception. I think in terms of luck, timing is critical for innovations. If the world isn't ready for your innovation, nobody cares. I think in this case, the work of the International Human Genome Project paved the way to make the world ready for this innovation at the time it happened. And you can't engineer that. Mm-hmm. That was just a consequence of the circumstances.
Yeah, I remember yesterday you said that an advice to young people that you have to fail quickly. That's a good one. To fail many times quickly and then you succeed. Right. Then the next one. When the first genome was published in 2000, there was much promised about personalized medicine that still has not arrived, at least in large scale. Do you think rapid sequencing has been the bottleneck, or are there some other bottlenecks, and what innovations would be needed to solve them?
So I think sequencing has been the bottleneck. I think the problem is most of us suffer from complex disease where lots of genes go slightly wrong. In order to identify the contribution of all those genes, you need to sequence a large number of people, and that's starting to happen. So I think it's happening, but we need to sequence lots of genomes to make this a reality.
I would like to add to that answer, if I may. So I agree with what David said, and I mean, in the examples we gave, cancer, rare diseases, and infection, these are real examples of decisions being made to help people today. But important to note that genomic medicine is just beginning. Mm. It's early days, and now in healthcare systems, certainly in places like the UK, it's being implemented. So physician, patient, genomic medicine. And it's going to take some time not only to build up the information, but to learn how to link that information to the kind of information that physicians normally record, and to also monitor the changing health of patients. So we need to be patient for another 10, 20 years before we decide on what genomic medicine can and can't do. This is just the beginning.
Yes, it's only 20 years since 2000. This is very short in this type of research. Okay, so you've been listening today, this innovation forum. So what key elements of innovation have you been hearing about that you think are more important than scientists realize? Was there something that struck you?
I think certainly for me, I think how we come up with these innovations, science is a much more social activity than people think. And I think in this age where people are rushing from meeting to meeting, we're not spending enough time talking to each other. And it's these simple things like going to the pub and spending some time with each other that are really important, and we're in danger of losing those. Mm-hmm. And that's what, in my opinion, drives innovation, social interactions with other human beings.
Yes, we agree. Okay. Now I would like to thank you once more for your wonderful talk and congratulate you again. And we will now have a message from the UK Minister of Science, Research and Innovation, MP, Amanda Soloway. So, Minister Soloway, welcome.
Good evening. My name is Amanda Soloway and I am the Minister for Science, Research and Innovation. It's a great pleasure for me to be able to join you at this prestigious event today. I'd like to thank Technology Academy Finland for organising this forum during these incredibly challenging times. As we've learned from the COVID-19 pandemic, innovation is crucial to an effective response to global challenges. The Millennium Technology Prize celebrates groundbreaking innovations that will improve our quality of life and promote a sustainable future. The UK has a strong tradition with this prize, World Wide Web inventor Sir Tim Berners-Lee, was the first ever Millennium Technology Prize winner in 2004, and Professor Stuart Parkin was awarded the 2014 prize for research that led to the phenomenal increase in the storage capacity of digital services, which herald the era of cloud computing. Today, I am delighted to congratulate winners of this year's Millennium Technology Prize, Professor Balasubramanian and Professor Klenerman, their teams and everyone they've worked with for their development of revolutionary DNA sequencing techniques. The next generation sequencing technology means that DNA can now be read in super fast time. This brings huge benefits to our citizens, from the fight against COVID-19, tackling cancer, to smarter food production through the better understanding of crop diseases. Using this technology to track and explore coronavirus mutations has already helped to create multiple vaccines and is critical to fighting new viral strains as they emerge. This technology will also prevent future pandemics. The UK has a long and proud history of invention that dates back centuries, from Ada Lovelace and Alan Turing, who pioneered early predecessors to computing, through William Grove, who invented fuel cells, to Frank Partridge, who helped save millions of lives by developing the first portable defibrillator. Breakthrough innovations like these need ambitious research funding. I'm excited about our latest initiative in the UK to launch the Advanced Research and Invention Agency, ARIA, a new independent research body to fund high-risk, high-reward scientific research. ARIA will be backed by some £800 million, which is more than €900 million in public funding up to the year 24-25. The funding will support the most inspiring inventors to turn their transformational ideas into game-changing technologies, discoveries, products and services. The COVID-19 pandemic has demonstrated the power and importance of global research collaboration, and this must inspire us to help address other pressing global issues, such as climate change. This November, the UK will be co-hosting the 26th UN Climate Change Conference of the Parties, COP26 in Glasgow. We must make Glasgow the moment the world comes together to solve the pressing challenge. I would like to close by thanking the Millennium Innovation Forum for encouraging amazing innovation with their work to stimulate collaboration. Professor Balasubramanian and Professor Klenerman and their colleagues for their development of revolutionary DNA sequencing techniques. And finally, you, the audience, for being innovators, supporters and advocates of innovation. Your commitment is helping the world to become a better place. Thank you.
Many thanks, Minister Soloway, for the greetings and the very inspiring words about the power of research. But now it is time for the Millennium Youth Prize. Inspired by the Millennium Technology Prize, Aalto University and the Academy of Technology are organizing the Millennium Youth Prize competition for innovators for the age of 19 and younger for the third time. This time, the competition invited young people to present solutions to global challenges on reducing food waste in stores, recycling battery materials and metals that are rare and harmful for the environment, and transferring different traits found in nature with the help of genetic engineering to benefit the world. This time we have a great opportunity for the winners, because the Millennium Youth Prize will be awarded by the Millennium Technology Prize winners. It's a big honor. So the Millennium Youth Prize jury, chaired by Professor Ossi Naukkarinen, Vice President for Research at Aalto University, has been doing its job today and made the decision among the 4 finalists on the winner of the Millennium Youth Prize. And now I have the great pleasure to reveal the winner of this year. The winner is a team of Aino Kuusi and Nikolai Argatov, and their project is titled Purification of Heavy Metals from Lakes by Bacteria and Algae using Magnetosome Gene Transfer. And here is how they describe their project themselves. The idea is to use genetic engineering in order to transfer the ability of magnetactic bacteria to form magnetosomes to other organisms. These genetically modified organisms would be applied to clean up polluted waters from heavy metals by bioabsorption. The genes would be transferred to endemic species in order to avoid the disruption of the ecosystem. After bioabsorption has concluded, the biomass would be harvested using magnetic and mechanical processes. And here is the assessment by the jury of this work. A special merit of the work is its imagination and innovation. It shows the ability to think out of the box. The phenomenon has been approached from an engineering point of view in a multifaceted way, so that different aspects of the problem work well. The work and discussion show an excellent understanding of the very theme of the challenge, the transfer of a gene to the technical scaling of its application. The work will also address its challenges and risks. The work is progressing boldly until concrete implementation of the solution. The best of the strong finalists as a whole. So, once again, big congratulations to Aino Kuusi and Nikolai Argatov, and may I invite them on stage.
Hello, do you hear us?
Yes, well, many congratulations on winning your prize. Maybe you could tell us why you chose this project and how you came up with your ideas.
Actually, we were doing the research for this competition because we were very interested over the years, because this third time this Millennium Prize has been held, and every year we were very interested, but just only this year we decided to participate, and we started to research into interesting phenomena in nature, and we came across these magnetactic bacteria. And at first we were coming up with solutions in medical applications, but as our knowledge grew, as our knowledge grew, there were already many different and unique solutions with using magnetactic bacteria. So we decided to, what if we look another way, maybe some ecological problems, and we came across the capacity to absorb heavy metals of these bacteria, and it grew from there.
Warm congratulations to both of you. I love the idea. I'm quite interested in, so you're a team of two people. So how did you divide up the tasks? How did you work together to have the idea? And how did you work together to go through all the possible solutions and come up with the one that you both thought was best?
Yeah, at first we just because of the pandemic, we were talking via some Discord and other on the phone, discussing different ideas and things we had researched online, and just brainstorming together. And it was, as you said previously, that the social interaction is very important, and we had some growing pains just because it was hard to, because we were very self-critical with our project and how the reality of it, and so it was at first a little difficult to work out solutions, but along the way we figured out some ideas, and it was.
Sounds like you
did. It was very, very interesting.
You did very well, considering the restrictions. and how we interact.
Can you tell us a little bit about your future plans and aspirations?
I just started my studies at Aalto University. I studied bioinformatics, so in the future I would like to maybe do some research work. Like, for example, your work is really inspiring and something that would benefit the health. And yeah.
For myself, I'm right now a student in Helsinki University in medical school, but during this year I realized that I miss math and physics quite a lot, so I'm in process to applying to Aalto University as well. So probably maybe try to study like in both medical school and some physics and engineering, and maybe find a way to like let them coexist together, maybe find some new interesting ways to do research or of some kind.
So I have a follow-up question. This is really to do with choosing what you might do in the future, what problems you may work on. So there are many important things in the world that need solutions. So how do you think about choosing which problem or which project you'd want to work on? What for you is a really good project?
For me, at least, I'm really interested in like medical applications and solutions, because they can improve the health of people. So, yeah.
For myself, and I think also for Aino, I think like our strength, part of our strength is to like we are very interested in biological applications, but we are also really like maths and physics.
Yeah.
So maybe like try to find something that really interests us, and we can use our strengths to solve problems to benefit the world.
Yeah.
Well, many congratulations on your award.
I would say the same. You know, you're in the most creative phase of your life. So please use that creativity. I'm inspired by what you proposed, and wish you all the best for the future. Congratulations. Very well deserved.
Thank you.
Thank you.
Right, thank you very much, and congratulations also from me. It's been a pleasure to hear about your work. I'm sure you'll go very far. I suppose I'm here to thank both winners, young and old, and older. Gentlemen, again, warm congratulations.
Thank you.
And excuse me, I'd like to thank also the chair of the session, Ilkka Niemelä, and Päivi, the chair of the International Selection Committee. We should take some time to reach some conclusions here and to think about the future. So for some concluding remarks, I'd like to invite to the stage Antti Vasara, the President and Chief Executive of VTT Technical Research, Finland. Antti.
Thank you, Jason, and good afternoon also from me. Yesterday and today have truly been inspiring days, I have to say. Millennium Technology Prize is Finland's tribute to innovations for better life, and we have certainly heard many examples today what companies, academia are doing to have impact on how society works, how economy works, and how we can live our lives as humans. I want to briefly recap some of the key points and my takeaways from today, and I've been helped by the rapporteurs, so many thanks then to the rapporteurs who have been invisible but working busily today. We started the day with a big picture. We were looking at both the grand challenges, but also then that how technology can solve them. And some of the things that I picked up from there were that there was, first of all, a very much a wake-up call for us in Europe. Europe may be off to a slow start in some of these later technology races, but it's a long race, like Karl-Henrik Svanberg said there in the discussion. We can close the gap if we want. Also, it's good to notice then that from Bengt Holmström's comments then that there's very different debate going on here in Europe than in US, that there's no room for complacency, as Nina Coppola said, that we need to raise the level of urgency, that we realize that the world is changing. Perhaps one of the things that we can use as our competitive advantage here in Europe is that we are very good at education. And that also applies for us here in Finland. And in the coming years and decades, millions and millions of people have to be upskilled, re-educated as jobs change and technology evolves. So maybe this can be then one of the ways that we in Europe can be successful. We need heroes, as Nina Coppola was saying. We need people who think big, who are willing to take the risks and chase their dreams and be entrepreneurs. And also, we need to realize then that we have a totally new type of asset these days at our disposal, and that's data. It can be used by millions, hundreds of millions of people at the same time, and it doesn't. It still is there. It doesn't wear down. It's very different type of asset that we have been used to, so that we need to be also thinking about differently than in the physical world. In the second session then, we had very much a future-oriented discussion going on, and some observations from there. Firstly, we have to take the time to think, and think slow and long term. This also came from Professor Klenerman in the discussion just a while ago. In crisis times, short-termism grows. And that is really then the enemy of thinking long term and thinking about solutions that are good also in the long term. We also need to pay attention to who is included in our futures, so that we're not only focused on the economical aspects, technology, these types of things, but we also remember that people are at the center of also future scenarios, and think about how to include as many as possible in thinking about and doing that foresighting work. And thirdly, connect the dots and challenge your assumptions. Don't just focus on one trend or the latest fashionable trend. Think about alternative trends, alternative scenarios, and also how things connect together. In the third session, we heard examples of then how companies and academia, researchers are turning science and technology to innovations with a real purpose and building better life. We heard examples ranging from very much on ground level all the way then to moon. So lunar cellular networks are even mentioned there, so that was high flying. On sustainability, we heard from Peter Vanacker that solutions for circularity and renewable solutions do exist today. Now it's time to put them into action. He very much gave great examples on circular bioeconomy. On the health, we heard how universal spare parts for humans are not quite here yet, but we're making already a lot of progress, and what an exciting future it can be then for treating many diseases and ailments. And for example, the group in Tampere University is already working on 3D spare parts or 3D printed spare parts for to correct vision. Then we heard on communications technology how that revolution that has already been going on for some time is now turning different industries into real-time industries and making agriculture smart, energy production cleaner, and bringing efficiencies into many other areas. And lastly, we were very much, I was very excited about these robogami, how to create new types of robots that are more personalizable and can be made to be useful in different types of solutions, even supporting different healthcare solutions and making better life for people. In the Millennium Technology Prize lecture, I find that whole story extremely inspirational. It is such a fantastic example of how you base something on really high quality academic work, turn that science into practical application, and then turn that into successful innovation, successful companies, but also then that having true impact in the world. As we have already seen that it has been helping, for example, now fighting COVID-19 and the pandemic in recent times. In today's different discussions, I think that the recurring theme has been this theme of curiosity, experimentation, and learning. Our modern life has been built on this through science and technology. And what a radical change has then happened in recent decades. On a personal note, I want to share one story. My own grandfather was born in the 1890s. And science and technology looked very, very different back then. Even Einstein was back then only a patent clerk and known for that. My grandfather was not a scientist himself, but he was intensively curious about technology and all the things new that were happening. Even when he was in his 90s, in the 1980s, he was trying to understand that what technology was there. He was always trying to use everything new, but all of that seemed like pure magic to him. It was a sheer miracle compared to what he was used to in his childhood, and he always wanted to understand things, but he never learned really those things. I have been there as a very self-assured teenage science nerd trying to explain to him how a C cassette player is working or how color television works, but I can tell you that it was not very, I never managed to explain those things to him. But he was in awe and admiration about all this. And today it would be so fantastic to discuss with him that what science has now made possible, and he would be full of admiration for the scientists and looking extremely positively and optimistically into the future. Will we feel that same awe and admiration in 2050? By that time, we have conquered climate change through the technologies that we're now working on. We have been able to eradicate many diseases. I am confident that we will have that awe and admiration in 2050 if we act now. As far as we are concerned, the most important time in history is going to be in the 20s, because this is the time that when we can act on science and technology and go and solve those grand challenges that we have been hearing about today. As Professor Balasubramanian said there in their talk, that opportunities like this are rare. Take them if you can. So we have hope if we want to see it. We have opportunities if we want to take them. Now is the time to act on science and technology and go make a better future for us all. I wish you have enjoyed today as much as I have. I wish you also share my optimism for the future that science and technology can bring. And I really thank you for participation. Thank you.
Antti, Vasara, thank you very much. A great summary of a great day. I should take this moment to thank all of our partners, all of today's speakers, all of the chairs of the sessions who I know have been working very hard to make this an engaging day of talks, and all of the rapporteurs who have been working behind the scenes to turn today into conclusions and documents that will be useful into the future. I think the future is perhaps the second most used word today after innovation. It's all been about the future. Briefly, thanks also to Eva Sievi, the COO of Technology Academy Finland, who's been doing an enormous amount of work behind the scenes also to make today great. But having mentioned the future, we'll move on to our final speakers. I'd like to call to the stage Markku Ellilä, the Chief Executive of Technology Academy Finland. Markku.
Thank you, Jason. And on the topic of thanks, I would like to extend a warm thanks to the organizing committee of this event, and especially its head, Professor Yrjö Neuvo, who has diligently and skillfully led the work leading to this event. I'd also like to thank all the speakers, all our partners whose names and logos you've seen in the background all through the day, all the people who have collaborated in making this event possible, and especially the staff that has been working for months to make this happen. And then special thanks also go to Professor Maria Makarov, who is now finishing her second 3-year mandate as the chair of Technology Academy Finland. I would like to extend our sincere thanks for her achievement over the last 6 years in further improving the quality and relevance of the Millennium Technology Prize. She will move on to another high-profile position, that of the president of the European Academy, Academia Europaea. The story of the Millennium Technology Prize continues. The next award ceremony will take place on the 25th of October next year, followed by the Millennium Innovation Forum on the 26th of October 2022. And we will be inviting nominations for this round starting the 2nd of August this year, and the round will close at the end of October. We are stressing that global issues require global solutions. Topics like climate change, loss of biodiversity, depletion of natural resources, and the pandemics you've heard many times during the day, they require our continued attention. And while technology shares some responsibility in many of these issues, breakthrough technologies will be a vital part of the solution. They will be enhancing our quality of life, they will be promoting sustainable development and improving diversity in its different forms. And the new thing in this round is that we also are interested and want to see that the ethical code of conduct is followed in the research that leads to these innovations. And to close my part, I would like to introduce the very last speaker. It is the next chair of Technology Academy Finland board, Professor Minna Palmroth from the University of Helsinki. She is a world-renowned researcher in the fields of space and high-performance computing, and she is highly active in societal impact, policy advising, and networking with the public and private sectors. Ladies and gentlemen, Professor Minna Palmroth.
Thank you, Markku. I would also like to start by thanking. I would like to thank Maria Makarov for being such an inspiring leader, and quite frankly, my role model and hero for many years. Without you, Technology Academy Finland would not be where it is today. As Markku said, my name is Minna Palmroth. I'm a professor of physics at the University of Helsinki. I'm also the director for the Finnish Center of Excellence in Research of Sustainable Space. This is essentially Finland's first own space program. Almost exactly today, at the end of May 2021, I have a 25-year-old career in space and high-performance computing. And Special highlights from this career are the 2 European Research Council grants which I have received. I am very proud and excited to be given this opportunity to chair the board of Technology Academy Finland. Science and technology are very prominent passions for me. This chairmanship gives me a very front row position, an opportunity to connect and combine science, technology, and business. This is something I've always wanted to do. In this position, I can promote 2 very important goals for Finland. On one hand, we can get the world's best brains to Finland every 2 years in the form of Millennium Technology Award winners. And I would also like to thank and congratulate Professors Balasubramanian and Klenerman for winning the award this year or last year. On the other hand, it gives an opportunity for the Finnish science, technology and business communities to actually connect and network with the world's best brains within the Millennium Innovation Forum. I am very happy and proud to start promoting innovations for better life. See you next year in the Millennium Technology Prize Award Ceremony 2022 and the connected Millennium Innovation Forum. Thank you.